Compared to genome sequencing, where the goal is to reconstruct the entire genome of an organism, in the next-gen sequencing, the goal is typically to use an existing genome and map many short reads to that existing genome. A metagenome is where you have a mixture of organisms in a cell culture, and that could be an environmental sample, it could be a skin sample where you have many types of bacteria, and you would want to identify which ones are present in that mixture. You would take the mixture and again shear the DNA, break it down to smaller fragments, and then read through your sequencing machine. So we have many different organisms here. We break down their DNA, we get many short reads. And then we would want to go back and label each of these short reads based on which organism they came from. So let's say this is my blue bacteria. These came from the blue bacteria. Okay. In order to identify which organism they came from, I would need a reference genome. So that blue bacteria needs to be well studied before, and its genome needs to be already identified. Okay. We'll say that's the blue organism. And the way I would go through these short reads is I would take each one, align it with the blue genome and the green genome and the red genome. So those are, say, 3 different types of bacteria, 3 different species. Okay. As I go through each short read, I'm going to ask, is it present here? Is it present there? Is it present there? If I find it present in blue, then I'm going to say that must have come from the blue genome, and I'll mark it as a blue organism. Going through all the short reads and mapping them to one of these 3 genomes would then tell me in the mixture what percentage of the bacteria is blue, what percentage of the bacteria is green and red. I would have to account for the genome sizes as well. So if the blue bacteria is itself a large genome, then breaking it down would result in many fragments. Okay. So I would have to normalize the number of fragments I see by the size of the blue genome so that it's not an unfair comparison when I try to recover the number of bacteria that are present in the mixture. So that's one thing I can do with a metagenomics analysis. Another thing I can do is in a single cell culture of the same organism, say a human cell culture, all the cells are of the same type. They're all human cells or they're all mouse cells. They've been grown in a well controlled environment. We know they're all the same cell type. Therefore, we know they are all the same genome. So we have one genome and a mixture of cells from that species. In this species, I would not really be interested in identifying the genome of the species because I already know that. Sometimes you may want to know what differences does this have compared to the reference genome. Reference genome is sort of an average. There's whoever they sequenced. So at the time where they had that genome sequencing project, whoever donated their genome to be sequenced, the letters represent their genome. Or sometimes it's going to be a mixture of individuals and you will get an average sort of genome, okay, the most abundant version of letters in each position. And then when you have a new person, you would want to know what differences does this person have from the reference genome. Those differences can be in terms of SNPs, which stand for single nucleotide polymorphisms. Say we have a short read that matches this position exactly, except there's a 1 character difference. That's a SNP. There are other types of variations, insertions, deletions, copy number variations for repeated patterns. Some parts of the genome contain repeated patterns. So say this is 5 copies of the same pattern. Some individuals will have more or fewer copies of that pattern. So copy number variation represents how many copies of this red pattern you have in your own genome. And how many copies of other repeated patterns do you have in your genome. So copy number variations is another type of variation from individual to individual. For the most part, our genomes are similar, up to 99.9%. Only a small fraction of the characters in our genomes are different. Another very important study that I can perform with this technology is, rather than analyzing the genome content, I can look at the RNA content. So if I capture all of the RNA molecules, for the most part, these will be messenger RNAs, but there are other type of RNA molecules as well. If we pull all of the messenger RNAs, collect them into a test tube, and add to that test tube the reverse transcriptase enzyme, I would need DNTPs because I'm trying to synthesize a DNA molecule, plus any other chemicals that this reaction would need. Reverse transcriptase will go through each of these messenger RNA molecules and use them as template and produce DNAs from them. Okay. So each one will now be replaced with a corresponding DNA molecule. And now I can take this DNA and feed it through my sequencing machine, which is really designed to sequence DNA but not RNA, based on incorporation of DNA nucleotides by DNA polymerase. So I need to start with a DNA molecule, which is why I have to take messenger RNAs and reverse transcribe them into DNAs. So we convert the problem of sequencing RNA into sequencing of the DNA problem. We know the sequences will map to our reference genome. Rather than just knowing what sequence we have, we are really interested in how much of each sequence we have in the RNA mixture. Okay. So some of the short reads may map to this region. This part of the genome is, say, gene A. And I have another part of the genome that expresses gene B, and it has fewer short reads mapped to it. Okay. So by counting how many times I have a short read that maps to gene A and how many times I have a short read mapping to gene B, I have determined the expression levels of these genes. So we can quantify the messenger RNA levels or the gene expression levels in a cell culture using this technology. A detail that I skipped is PCR here. You would usually take your sample, which has a small amount of DNA content, and you would replicate it to increase the abundance of DNA using polymerase chain reaction, which is essentially a DNA synthesis in repeated cycles. Double the amount of DNA, and then you quadruple, and then times 8, times 16, however many times you need. Okay. So this is an optional step. Depending on the sequencing technology you use, you may need to do that or not. Okay. There are some computational challenges here, though. The main computational challenge is, after we identify a short read, finding out where it is located in this genome. So you're going to have to find this read in the genome. I'm going to say the genome is 1 gigabyte, which is 10 to the power 9. And each short read is, say, 100. So to search this one against the genome, you need roughly 10 to the power 9 times 10 to the power 2 amount of time. And then you would have to repeat this for every single one and identify where they came from. So let's call that n. The number of short reads is in the order of millions or billions. So you're going to have many millions of these. If you use a local alignment method or a global alignment with free end gaps, you would have to pairwise align each short read against your genome, and that is n times the length of each short read times the length of the genome. That's how much time roughly you would have to spend. In summary, it's just too long, right? You wouldn't want to spend that much time. If this were a human next-gen sequencing study, using alignments of each short read would probably take you days to complete. I can use BLAST, right? I can take this, that's my genome. I can create a BLAST index from it, and this creation needs to be done once. Once I create this index file, I can reuse it as many times as I'd like for each short read. I'm going to pull out this short read, search it against this index, and I will more or less do this in an amount of time that does not depend on the size of the G, roughly speaking. So I've reduced the problem to run in time n times the length of each short read. So each search will be fast, but you have many of them to perform. The methods that I will discuss today are designed to deal with this large N. Can we do something to improve this BLAST phase and develop a strategy that is better than BLAST? The assumptions that we are going to make will make our strategy not as powerful as BLAST in terms of the search results, but it's going to be better suited for the problem that we are solving. We typically use BLAST to do homology searches, where the read or the query that you search against your database and the hits that you find are going to be slightly different or very different. But in a mapping problem of short reads to a reference genome, we know things should align very well. So the number of mismatches is going to be very minimal. For most of them, it's going to be identical hits. For some of them, there may be 1 or 2 characters different. So BLAST is sort of an overkill for this next-gen sequencing. We would not really want to do BLAST. Because of the overkill, it takes more time. It tries to find sequences that are not just near identical, but also similar. We don't need that much similarity. We only need near identical hits. The first method I will describe is suffix trees. I will describe the methods in terms of finding exact matches. I will try to find places in the genome without any mismatches. There are extensions of these methods that allow a few mismatches, say up to 3 mismatches, but I'm not going to really discuss those in detail. Let's discuss the problem of having a short read and finding where it is located in the genome, finding an identical match without any differences in the genome composition. So no substitutions, no gaps. The first method is suffix trees. And the analogy that I'm going to use is where the pattern is a car, and I'm searching it in the genome. Panama bananas is the genome, and the short read that I have is Nana, and I'm trying to find where it is located. This string search is a common function in most programming languages. In MATLAB, you would use strfind to do this. You would say, go to G, find where P is located. What strfind will do is it will take this pattern or short read, and in a for loop, it will ask, does it start in the first position of the genome? No, it doesn't, because there's a mismatch in N. And then it will ask, does it start at the second position? No, it doesn't, because N doesn't match with A. And then it will ask, does it start in the third position? This part is good, this part is good, this part is not. Let's think about the number of operations that we perform. I would have to try out every single position as a candidate starting position for my pattern. Roughly speaking, the last few I'm not going to use. I won't have enough characters left to complete the search, but I'm going to ignore that last few. I'm going to approximate this as G. So there are G positions that I can possibly have a hit. When I inspect a position, I need to compare with every character in my pattern. So I need to compare up to 4 characters. Only when all 4 of them are equal, then I would say, okay, this is one answer. I would need a for loop that goes over each of these characters and checks to see if they are the same as between the pattern and the genome at that position. So that's G times P. taking this pattern and driving it along your genome would take in the order of G times P amount of time. If you use a pattern that's twice as long, you would spend twice as much time to find it. If you use a genome that's twice as long, you would have to spend twice as much time to find the pattern. If they are both twice as long, then you would spend 4 times as long to find the pattern in the genome. So that demonstrates the process. And the car drives along, finds where it is located. Now, in the next-gen sequencing, we can certainly perform this strategy where we have each short read and we can drive it along the genome, find where it's located, and then take the second pattern, drive it along the genome, and find where it is located. So find that, find that, find that, find that. If the number of patterns, number of short reads is n, that means I would have to perform n times g times the length of the pattern. If I have twice as many short reads, I would have to spend twice as much time. To find a single pattern takes this much time. Here, text is the genome size, pattern is the pattern size. When I have multiple patterns, this is really n times pattern. Total length of the patterns is going to be this. So if you merge all the short reads and ask how long is that, that's going to be the number of short reads times the length of each short read. In a typical next-gen sequencing for human genome, the size of the genome is in the order of billions. The total size of the short reads that you get from the sequencing machine is in the order of 10 to the power 12. That's a huge number, right? This complexity is not going to work well when we try to run it on our computer. It's going to take a long time. One idea is to pack all the short reads into a bus that we will drive once. Okay. So as we drive the bus along the genome, we want to be able to discover for all short reads at the same time, whether they start at that position of the genome. I can create a tree structure that merges, that combines all the patterns into a single data structure that I can then simultaneously search in the genome. Here's how I'm going to create this data structure. I'm going to go through each short read, each pattern, and add it to the tree. I've added banana. Next, I'm going to add pan. Right? That's the next pattern that I would like to find within the genome. Next, I'm going to add and. Okay. Next, I'm going to add the next one, nab. The next short read is antenna. Okay. When I add antenna, I'm not going to add it separately. Instead, I'm going to follow the common branch that it has with another pattern, A, and then N, and then I'll split it into T, E, and NA. Okay. So I'm sharing the commonalities between patterns when I create this tree structure. So I've added antenna. When I add bandana, I'm going to go down band and then split into dana. Okay. And similarly for the other patterns. So I have packed all the patterns into a bus, into the same data structure. We're going to call this a tree or tree. As we drive this bus along the genome, we can simultaneously search all of the patterns. Here is the algorithm. We're going to drive this tree along the genome, and for each position, we're going to walk down by spelling the symbols of the genome. If we end up at a leaf node, that means at the position that we searched in the genome, there was a hit for this pattern. If we get stuck, that would mean none of the patterns start at that position. We're going to move on to the next position of the genome. Let's demonstrate that. So this was my genome. This is the collection of all the short reads that I'm searching. I'm going to try the first position of the genome first. I'm going to go through every character here and spell out P, and then spell A, spell N. I ended up at a leaf node. I have found out that there is a hit in the genome for this leaf node. Whatever pattern was used to end up at that leaf node, there's an occurrence in the genome of that pattern. I'm going to move on to the next character. And I'm going to spell out A, go that way. N, go down. A, go this way. M, I got stuck. So there's no hit starting at this position. None of the patterns that were used to construct this tree are present starting at this position of the genome. I'm going to move on to the next one and repeat the process. If I get stuck, I will declare that there's no occurrence. If I don't get stuck and successfully get to a leaf node, that would indicate the presence of that pattern in the genome. And so on for the remaining locations of the genome. So how much time does this gain us? Let's remind ourselves that the complexity of the naive approach of the simple solution of driving every car separately was genome size times the size of each pattern times number of patterns, or if we combine these two, we would say the size of all the patterns. When I'm inspecting this option, I'm going down the tree. The number of times I go down and match in the tree will be bounded by the length of the longest pattern. The longest pattern is this one. That's 1, 2, 3, 4, 5, 6, 7. So no matter which of these positions I'm currently checking, I know I would not have to compare more than 7 steps. And there are G positions that I would have to try. Therefore, the complexity of this pattern tree is the size of the genome times the size of the longest pattern. So I have gained a lot. The size of all the patterns was 10 to the power 12. That's the typical size. The size of the longest pattern is only 100. So I have made a gain in the order of 10 to the 10. If this one takes 1 second, this one takes 10 to the 10 seconds. I've drastically reduced the amount of time I would have to spend for searching all these patterns. There is, of course, an initial investment in terms of creating this pattern tree, but that is negligible compared to 10 to the power 10. In addition to time complexity, which represents how much time I would spend searching, I also need to consider the space complexity, which is how much memory or how much hard drive space am I using to solve this problem. The space complexity of the tree implementation will be bounded by the number of edges I keep in the tree. And that's in the order of the collective size of the patterns, which is 10 to the power 12. That's still a large number. So I have gained in time, but I would have to spend a lot of memory to solve the problem. This is not too bad because I already need to have a storage for them. The size of the tree is just, in a way, doubling the amount of storage I already need, which is not that bad. In packing the patterns onto a bus approach, I would have to repack new patterns for every experiment I perform. So if I take one cell culture and run next-gen sequencing and get my short reads, I would have to pack them onto the bus, create a tree structure, and then search them in the genome. Tomorrow, when I run another experiment, I would have to pack those short reads again and repeat the search again. If I can find a way to pack the reference genome instead, I would only do that once, and no matter how many reads or how many new experiments I perform, I wouldn't have to repack them onto the bus because I'm not going to use that strategy anymore. I will maintain the genome on the bus rather than the short reads on the bus. To pack the genome onto a bus, what we'll do is we'll create all suffixes of the text. With short reads, we saw a way how to add them into the tree, right? The genome is one sequence. If I use the same strategy to add that to a tree, it will just be one giant branch. And that's not going to be helpful in terms of searching. What I need to do is use all possible suffixes of this genome and create a tree from them. And in a way, I'm packing all possible starting positions that I would need to search in the genome onto a tree structure. Exploring that tree structure later will mean that I'm simultaneously searching every single position of the genome at once. So when I go down the tree, I'm simultaneously exploring many different places of the genome. So rather than walking or driving the car linearly, I'm simultaneously sort of exploring or searching many different positions of the genome. This is one suffix. Another suffix is this one. Another suffix is this one. And this one, and you get the idea. And I'm going to add each of these to this tree. So I'm going to do P, A, N, and then all the way down to M, A, B, A, N, A, N, A, S. When I add this one, that's going to create an A branch. And then M, A, bananas. I do have the tree, so I'm just going to show that to you. I'm adding every suffix of this. Whenever there's a shared branch, I just reuse that shared branch. The $sign is a special symbol that I need to add to mark the end of the text. It marks the leaf node, just tells me where the end of the genome is. Let's say the end of the genome was AB. Pretend that this is the tree that I've created up to this point, and the last two characters I need to add is AB. I would have to go to A, B, but there's already something else here. I would have to invent a way to mark that as a leaf node as well, but it's not really a leaf node, it's an internal node. By adding a special dollar character that is going to be a character not present in the rest of the genome. So this needs to be a character not used in the rest of the genome. So adding AB, dollar would then mean I would need to add A, need to add B, and need to add a $sign. So the $sign helps us avoid the problem of a suffix being embedded and hidden as an internal node. Adding the $sign makes sure that each suffix ends with a leaf node. So now let's talk about how I can search a pattern, a short read, within this tree structure that I created from the genome. I'm going to search the word Nana. I'm going to drive Nana along this tree. I have to go down this way. So I drove to N. Next, I need to drive to A, and then NA. When I drove the car over here, that N is representing that sequence, this suffix, and this suffix. Okay. So driving the car just one stop helped me explore 3 different positions of the genome simultaneously. Driving the car one more time to the letter A simultaneously drives on the genome to these characters. driving the car one more step to a letter N, simultaneously drives in each of these positions. So this one doesn't match, so we're not going to drive that anymore. This one matches, we're going to drive that. This one doesn't match, we're not going to drive that. So driving on the tree essentially means we are driving along these three paths simultaneously. Taking one more step takes a step there, a step there, a step there. Taking one more step would go here, fail, which means we're not going to follow that route anymore. This one is still good, this one fails, we're not going to follow that route anymore. And then one more step goes here, and we have found our hit. We have found where Nana is located. Searching this Nana down there, we go down. All right, we found a match. Let's talk about how long it took us to find a match. The number of steps that we would have to travel down is just the length of the pattern. Let me use a small p for that. If I'm searching for Nana, I just need to go down 4 steps. I don't need to explore the rest of the positions of the genome. So in all of the previous solutions, the size of the genome was part of the complexity. In this new approach, the amount of time that I would have to spend to find a short read is just the size of the short read. So this is, again, a tremendous amount of saving that I would have in searching a short read against the genome. Creating this suffix tree, of course, would take time. So I would need an initial investment of taking the human genome and creating the suffix tree structure from it. But that can be done only once. I can reuse it anytime I do a short read search. If I need to find banana, I go down to banana. If I want to find nab, I do that. If I want to find antenna, I go down there. Whenever there's a mismatch, I just stop and say there's no match for antenna. So far, what we have done is discover whether or not the pattern is present, but we haven't really talked about where it is located. To find out where it is located, what we can do is when we add each suffix to this tree, we can mark the position in the genome that gives rise to the suffix. So this one is the first position because it's the Panama bananas. This one is the second position. I'm just going to mark the leaf nodes. And then what's the next largest one? Third position, 4, 5th, 6th, 7, 8, I think 9, and so on. Okay. When I created every suffix from the genome, I would also mark what was the starting position and then add it to the suffix tree and label the leaf node with that starting position. So I have to remember what was the starting position for that suffix that I added to the suffix tree. When I perform a search, and let's say I find banana, which means I stop the car there. After I discover that the pattern is present, because I was able to match with every character of the pattern, I have to keep going down this path all the way to a leaf node and look at the number for this. I think this was supposed to be 6. And that tells me what the starting position of this B is. The leaf nodes represent the starting positions of the suffixes that gave rise to that path. So the idea is after we've matched with our pattern, we're going to keep going down to the leaf node and use that number to report the position of the hit. What if we end up at a place with multiple hits? Let's say I'm searching for the pattern Anna in the genome. I would have to go down here, go down there, and stop here. So I know there's at least one match, but where is it or where are they? What I would need to do is go down all the paths following that branch. And these are going to be the positions that the pattern ANA is present in the genome. And you can double check. 179, that's 1. I think they are 0 base for this example. This one is called 0. The hits for ANA, there's 1 here, there's 1 here, and there's 1 here. 1, 7, 9. So we find a match. We go down all the paths, the leaves, which contain the recorded locations of those patterns in the genome, those suffixes in the genome. We have made significant gains in time. Let's talk about the memory requirements. We would have to create all suffixes of the genome. The number of characters is essentially the size of the genome squared, roughly. So the memory footprint is OG squared. If the size of the genome is 10 to the power 9, we would need 10 to the power 18 amount of space in the suffix tree of the genome. That's too large. We would want to keep the suffix tree in memory, in RAM, in order to perform quick searches. Right? We want to be able to take each short read and drive it through the tree in the memory. So what we would do to optimize this is the following. We don't really need to keep all of these characters or all of these nodes. We can collapse them into a single text. And likewise, we're going to collapse all the other branches where there's a single path. And anytime there are no branches, you can collapse them into a single edge. So this is a condensed suffix tree. Furthermore, you also don't need to keep these texts that are already present in the genome. For example, this text is going to be the entire genome itself. So if your genome is 10 to the power 9, this would be a text that's 10 to the power 9 long. And we would not really want to keep yet another copy of the genome. The entire genome will be in one place. What we can do instead is say that this branch starts at a particular location of the genome and is this long. We're going to replace that bananas with the starting position within the genome and the length in the genome. We're going to do that for all the other texts as well. That way we are avoiding keeping these texts in memory. They'll just each be 2 numbers. So significant space savings. And therefore we have really reduced the space requirement from G square down to G by condensing the branches and also keeping the locations rather than the branch sequences themselves. Let's look at an overview of the complexity for the suffix tree. It allows us to do fast, exact matching of the patterns or finding the patterns. The time requirement is the length of the genome plus the total length of the patterns. The memory requirement is the length of the genome. Even though this is small, there's some overhead in the data structure itself. So roughly, you would need 20 times 10 to the power 9 amount of RAM. So 10 to the power 9 is 1 gigabyte in a way. So this would mean that you would need 20 gigabytes of RAM in order to store a suffix tree of the human genome. That sort of pushes the boundaries of a typical laptop. If you are using a desktop, it should be possible. Otherwise, you would have to keep it on the hard drive, which is smaller to access. You would lose in time if you cannot store it in the memory. The construction of the suffix tree can be done linearly in the size of the genome. The suffix trees are nice, but we need a better method that has a smaller memory footprint so that we can run it on our laptops. And we also need a way to handle mutations. The suffix trees are not so great at handling mutations, although there are some extensions to allow mismatches, they are not as trivial. So we want to be able to do approximate matching with a smaller memory footprint.